So we will continue from where we have stopped. But before that, I would like to uh, rebrush your memory with respect to what we have discussed in the previous class. We are discussing about the muscle contraction, and before that, what's important for us is uh, the structure of the muscle fiber and how it is important for us in understanding the process of muscle contraction. So, in a muscle contraction, the mechanism in order to understand, there are different models, out of which the most accepted and well defined model appears to be sliding filament theory. If I want to explain what exactly meant by the sliding filament model in a nutshell, it is nothing but the sliding over of actin and myosin filaments, and which brings the contraction. Once they slide over, that becomes the contraction, and once they stop sliding, that is the relaxation. So this is the uh, principle that is lying behind this model. So hence the name sliding filament model. So usually what happens is muscle shortening occurs due to the movement of actin filament over the myosin filament. Myosin filaments do not move. It is the actin filament. But myosin filament makes the actin filament to slide over it. And how it is formed? It is made possible by someone. Uh, this is achieved by developing the cross bridges between actin and myosin filaments. So this is as a result, what happens? The distance between the two jet line of the sarcomere decreases. All of you know, one sarcomere uh, is nothing but the two the area between two jet line. That what we call the sarcomere. So now um, here what happens is ultimately excitation contraction coupling. What do you mean by this excitation contraction coupling? This is the mechanism by which action potential is generated, and that causes this myofibrils of the muscles to contract by sliding the actin filament over the myosin filament. This is what we call excitation. Excitation, it is just to generate the action potential. Contraction coupling, because there is a coupling of both actin and myosin with the help of the cross bridges, and thus it brings the contraction of the entire muscle fiber, that is one cell. So here again I would like to draw your attention about the structure of myosin and actin filaments, that is the thick filaments and thin filaments. So here in the thick filaments, the myosin, the it is having a head, it's having a double head, and which is very very essential for the development of the cross bridges. So this is this head which I am showing with the cursor can bind with the active site on the actin or on the thin filament, and here it bind, and then when along with the ATP, when the ATP hydrolyzes, the binding of this myosin head with the active site on the actin releases. This is what. So actin has, if you see this, this is the actin filament which is having myosin binding sites. But when the muscle is in rest, these actin filaments are not exposing the binding sites, because these active sites are masked by a special protein what is known as tropomyosin. So when the muscle fiber is in rest, these active sites are masked by the tropomyosin protein. So this tropomyosin protein, uh, when the cell has to contract, now what happens? The tropomyosin is removed by some enzymes so that the active sites gets exposed, and so that to the active sites the heads of this myosin filament can attach and thus form a bridge. This is the uh, principle that is underlying that. See here the actin filament. So these are all the active sites. But generally, the active sites are blocked when it is in rest uh, by the protein what you call the tropomyosin. It forms troponin complex and then binds them. So especially when the calcium ions combine with the troponin C, the troponin complex is it undergoes conformational changes in some way so as to uncover these active sites. Active sites are uncovered when the calcium ions combine with this tropomyosin C, and which makes some structural changes, conformational changes, and thus it uncovers what we call these active sites. And when the active sites are uncovered, then like this, then um, the myosin head can bind with this. Somebody I have to admit. Uh, See, this is how the interaction happens. The interaction between the cross bridges and active filament. It is causing the contraction. See here, you can see how the heads of the myosin have attached with the active site on the actin filament, and that not only attaches. After attaching, they tilt the head and so that they move these actin filaments, both the actin filaments together, thus makes this actin filament to slide on this myosin filament. See, this is the arrangement of these thick and thin fibers. That is the myosin and actin fibers. This is one sarcomere. This is the jud line. The space between these two, it is known as the sarcomere. And this jud line or jud disc. Now, see here, the actins and myosin they are arranged alternately, but leaving a very large gap. Now what happens? The heads of these attaches here and pull this uh, actin filament over the like this. See here you can see in a relaxed state how it happens. This is the jud line, and in the contraction state, what happens is this binds with this. The myosin head joins the actin filament and pull the jud line on both the sides together, thus decreasing this central area which you can see. Now, in a fully contracted one, there is no this central area, what you call this thick uh, bare zone, what we call the bare zone will not be there. See, the actin filaments slide over the myosin, and they also slide over the other actin filaments also. See now as a result what has happened, this is now see this is in the first relaxed muscle state is the length of one sarcomere, and in the fully contracted muscle you see the length of one sarcomere. The entire length of the sarcomere, that is the length, these two jud lines are brought or pulled together, thus decreasing the area of this. That is what happens in this sliding filament theory, how the muscle contraction. Now, what I will do is I will try to explain this entire process of muscle contraction step by step. The first step for the muscle contraction to happen, there must be an action potential, which is caused by the depolarization. Now, all of you know what is the action potential. It this action potential will come along the motor nerve because all these muscles are connected with the nerves. The information should come from the brain or the spinal cord or in general what we that is action potential travels along the motor nerve from the along the central nervous system. And this information through the nerve endings enter into the muscles. So this action potential through this motor nerve endings reach the muscle fiber, indicating or requesting the muscles to contract. And at each ending, you can see each ending of the nerves secretes small amount of neurotransmitters. Because while studying the nervous system, we have seen that the nerve endings are having the vesicles which are enclosing the neurotransmitters, especially the acetylcholine is the major neurotransmitter. A small nerve secretes a small amount of this neurotransmitter, that is the acetylcholine. So now, actually, where the nerve meets the muscle, again there is a synapse is formed. This is, we do not call it as synapse, but it is neuromuscular junction. It is called as neuromuscular junction. There also there is a small gland, that is neuromuscular cleft. We call it as that is the neuromuscular cleft. So now acetylcholine, that the neurotransmitter, is released from this nerve ending on the local where it has innervated on the muscle. That is on the local area of the muscle fiber. So now what happens once uh, the acetylcholine reaches the muscle membrane, muscle fiber, it opens multiple gates known as acetylcholine gated cation channels. It opens number of acetylcholine gated cation channels, because these cation channels are again, there are protein guards are there, and through the protein molecules floating in the membrane, and that opens this acetylcholine gated channels. See, again I will repeat, action potential travels along the motor nerve, and at the nerve ending there are neurotransmitters, and these neurotransmitters are released, and this released neurotransmitter, that is the acetylcholine, acts on the local area of the muscle fiber and makes this acetylcholine gated cation channels with the help of the proteins. It makes it open. Action potential, I have told, the rapid rise and subsequent fall in the voltage or membrane potential across the membrane in a characteristic pattern, which I have already told you. Again, I am not going to repeat that. So now, the opening of this acetylcholine gated channels, what makes it allows large quantity of sodium ions to diffuse into the interior of the muscle fiber. Once these acetylcholine gated channels are opened, there is an influx of large quantity of sodium ions inside the muscle fiber membrane or interior of the muscle fiber membrane. This causes the local depolarization. Because of this local depolarization, voltage gated sodium channels are open. See, this initiates the action potential at the membrane. See, this is the step. When the acetylcholine gated channels are opened, large quantities of sodium ions enter into the inside the membrane, muscle membrane. And once these large quantities of sodium enters inside the muscle, which causes the local depolarization. As a result of this, voltage gated sodium channels are open. Once the channels are opened, this initiates or it starts the action potential at the membrane. Once this action potential is generated, the action potential always travels along the muscle fiber in one direction. At the same way, it travels in nerves. So now This action potential depolarizes muscle membrane, and most of this action potential, again it's voltage gated, that the electrically flows through the center of the muscle fiber, and here it makes the sarcoplasmic reticulum. I have told you sarcoplasmic reticulum is nothing but the endoplasmic reticulum, which is the storehouse of calcium ions. It makes this sarcoplasmic reticulum to release large quantities of calcium ions, because these calcium ions are stored in the sarcoplasmic reticulum. Now, these calcium ions which are released, they develop a force between the actin and myosin filaments, and so that the myosin heads can bind with the active sites on the actin and slide alongside with each other and pull the actin filaments together. This causes this contractile process or contraction. All these things happens in milliseconds. Immediately after that, calcium ions are pumped back into the sarcoplasmic reticulum, because there is calcium ion membrane pump. This calcium ion membrane pump these calcium ions back to the sarcoplasmic reticulum until new action potential comes along. So this removes the calcium from the myofibril, and as a result of that, what happens is the attraction force between the myosin head and actin filament reduces, and the muscle enters into a relaxed state. See, here again I have shown you, this is the relaxed state, this is the contracted state. Sliding filament, see the distance of the Z line, it decreases. Uh, Actually, the sliding mechanically, if you want to study, it is caused by the force generated by the interaction of the cross bridges. So these cross bridges, they pull them. See here, you can see this is also known as walk along theory. Interaction between activated actin filament and myosin cross bridges. So this is the myosin. This is the active side. This is the myosin. It attaches, pulls, and releases. Pulls and releases. So this is known as walk along. It is just like walking. The head attaches on the active side. As a result, a force is caused between the head and arm. Now, once they attach, the head tilts towards the arm. The head tilts in this way. And as a result, they drag the actin filament along with that. And this tilt of the head is called as power stroke. They attach. See, this is first straight. See, when it is tilted, it bends towards the arm. So this is called as, the tilt of the head is called as power stroke. Then immediately after tilting, the head automatically breaks away with the active side of this and return to the extended direction like this again, and attaches with the new active side. Again tilt, detaches, extend the head, attaches with the another active side, and tilt, and power stroke is created. New act further down along the filament, and uh, as a result, the active filament moves step by step. But all this process happens so swiftly, accurately, and uh, fast that there is no gap or lag. So that is done simultaneously. So the heads of the cross bridges bend back and forth, step by step, just like walking. And uh, So here the head of the cross bridges should bend back and forth, step by step, and this is like just walking along the actin filament, pulling the ends of the both the active filaments toward the center of the myosin filament. This brings the contraction process. See here, this is the myosin head attaches, and usually at the beginning of the cycle, no ATP is bound to myosin, and myosin is tightly attached to the actin. When the myosin head is tightly attached to the active site on the actin, this position is known as rigor position. This position is known as rigor position. In rapidly contracting muscle, this state is very brief in milliseconds. Then the ATP attaches here. Initially, ATP will not attach. When the myosin head tightly binds with the active site on the actin, this is known as rigor position. Now the ATP attaches. Otherwise, if the ATP doesn't attach to the myosin head, this rigor position will be permanent. It is not going to detach from the active site of the actin. So that is known as rigorous mortis. I will come to that later. See, the ATP binds to the cleft on the myosin head now, and this brings some conformational changes on the myosin, and that decreases the affinity of this myosin head with the active site on the actin. As a result, myosin is released. This is possible because of bringing some conformational changes in the head of the myosin. This takes place by the attachment of ATP. And once the attachment is detached, the position is no more called as rigor position. See, what upon the cleft? Now, once it is detached, the cleft closes around the bound ATP molecule and brings further conformational changes, makes the myosin to be displaced toward the plus side. Usually, this is the minus end on the actin, this is the plus side. Earlier, it was bent for stroke. Afterwards, see, this becomes stretched towards the positive end. And ATP is hydrolyzed to ADP and phosphate, uh, and that may remain attached with the myosin. But now this head has detached and it has displaced towards the plus end or positive end. Now the myosin head bends with the new active site on the head towards the plus end, and again the power stroke is formed because the head tilts like this toward the negative end by with the attachment. Then ATP attaches, brings conformational changes. Then the affinity between myosin head and active site decreases. The head shifts more towards positive side and finds out some more active site somewhere here and attaches. So that's why it is just like walking from here also the active first it has attached somewhere here, then it has attached with this active site, next it will attach one more active site, then so it is the head is just like walking on the active sites of the actin filament. The ADP release um, again the ATP is hydrolyzed to ADP and phosphate, and this ADP is released and myosin returns to the original state. But the cross bridge cycling continues with the myosin toward the plus end of the actin filament. How long? As long as the calcium is bound with the troponin C. because when the calcium is bound with the troponin C, the tropomyosin unmasks the active sites. But once the calcium fail to bond with troponin C, then tropomyosin will cover the active sites and the myosin heads no more can contact the active sites. So the relaxation occurs when calcium reaccumulates in the sarcoplasmic reticulum um, by the with the help of an enzyme calcium ATPase. Now the tropomyosin cover the active sites. This is what we call the relaxation. See, this is the cross bridge cycling. The cycle stops here and relax, leaving muscles here. Then see here, myosin heads develop the cross bridge. They bind. Then they bind and then they pull them forward. Then ATP attaches, brings the conformational changes, detaches. Then ATP gets hydrolyzed into ADP and phosphate. And then the myosin head attaches with the new active site, tilts its head, brings the further contraction. Again, ATP binds, detaches with the actin, hydrolyzes, search for next active site. This is how these cross bridges are formed. Here I would like to introduce one more terminology, that is rigorous mortis. This is observed after few hours of death, usually 15 to 25 hours of death. What happens is all the muscles of the body go into the state what we call contraction. All muscles will contract, all muscles after the death. This is called as rigorous mortis because muscle contracts but never relaxes. Because there is no, even without action potential, what happens is calcium ions are released, they attach with the troponin C, unmask the tropomyosin, and the myosin head binds with the actin site. But there is no loss of ATP because ATP will not attach. As a result, the attachment, the rigor position is the one when the myosin head attaches strongly with the active site on the actin. The rigor position will remain as it is. It will not undergo relaxation. How long? Till the muscle proteins start deteriorating. Again, it may take 15 to 25 hours later because of the autolysis which has been caused by the lysosomes. And this, all of you might have seen the cadavers or dead bodies. All muscles are in a contracted state. And that is known as rigor mortis. Because only contraction happens and this contraction is a permanent feature. So that's why this is known as rigorous mortis. Now, this is how the contraction and relaxations are brought about. Now, what's important is, like we have seen in the last class, while discussing the nervous system, we have seen something about the synapse. But here also, the same types appears in muscles also, which is popularly called as neuromuscular junction. This is the site where nerves communicate with the muscles, thus forming a junction which is known as neuromuscular junction. So this neuromuscular junction, how it is appears, see, it is the synapse. It is also nothing but a synapse between the motor neuron and muscle fiber. Where the motor neuron communicate with the muscle fiber, we call it as neuromuscular junction. What are motor neurons? The nerves that innervate the muscles and bring the information from the central nervous system with respect to the action that is to be carried out by the muscle. One motor unit refers to single motor neuron and the muscle fibers it innervates. Single motor neuron and the muscle fibers may be more, that it innervates. See, this is how the neuromuscular junction appears. This is the neuron. This is the neuromuscular junction. This is the motor neuron fiber, and at the end it branches. All nerves they form the branches, and here you can see the muscle fibers and the nucleus also, and the end of the nerve fiber branches you call it as new motor end plate. This is the motor end plate which communicates with the muscles, and here lies, below this lies the myofibril of muscle fiber. If you analyze this junction, it will appear like this because the nerves at the terminal end, it forms number of branches. This is one branch and this is one muscle fiber in which it innervates. See here in both the, this here there are vesicles. Here they form a bulb-like structure. In that they are rich in mitochondria. Here in the muscle also in that area they are rich in mitochondria, and this is what we call synaptic vesicle. This entire this thing. A synaptic vesicle communicates with the muscles, but there's a gap. But there's a gap. This gap between the synaptic vesicle and the muscle fiber, synaptic vesicle and the muscle fiber, the gap is known as synaptic cleft. Then in the muscle fiber, there's a folded sarcolemma. That is the muscle memory, and below that you can call it as the motor end plate. So neuromuscular junction is the site where motor neuron meets the muscle fiber. So it is separated by a gap. There is also a gap like the synaptic gap. Here you have neuromuscular cleft. Another terminology, that is the motor end plate. Motor end plate is nothing but uh, the pockets that are formed around the motor neuron by the sarcolemma. The sarcolemma, the plasma membrane of the muscle, it forms pocket around the motor neuron, especially around the neural vesicles, and that is known as motor end plate. And actually, the acetylcholine or neurotransmitter is released from this motor neuron. Once the neurotransmitter is released from the motor neuron, it causes end plate potential. That means EPF, nothing but depolarization of the muscle fibers. If you study the physiology and anatomy of neuromuscular junction, an axon approaches the muscle. Where the axon approaches the muscles, it divides into many terminal branches. And at this terminal branch, there is no myelin sheath, you remember. Otherwise, nerves are coated with protein, what is known as myelin, and It covers that. But in this region, there is no myelin sheath. Each of these neuron terminal forms special junctions, either with one muscles fiber or with more number of muscles. A single motor neuron can form number of branches at the terminal end, and each branch can innervate different muscle fibers also, or they can attach with the same muscle fiber depending upon the condition. See here, this is the motor neuron and they branches. See, these are the different muscle fibers that they innervate and they form terminal buttons. Where these terminal buttons attach the muscle fiber, you can see the neuromuscular junction. See, these are the terminal buttons which fit into the shallow depression. Because on the muscle fiber also there is a shallow depression where on this muscle fiber there is a shallow depression. These buttons can perfectly fit into this shallow depression just like lock and key. And underlying muscle fiber, these are the enlarged. These are all the neuron vesicles. See how these neuron vesicles have a vesicle-like shape which can perfectly fit into the depression on the muscle membrane. So now what happens here? The See, this is the neuron. Myelin sheath ends here because in the branches there is no myelin sheath, as I have already told you. The action potential is propagated along the motor neuron, carrying the information, and that comes to the terminal button. This is the terminal vesicle or terminal button. In the terminal button, you can see here there are action potential is generated because there are on the muscle fiber because there are sodium gates as well as calcium channels which you can see. Now, when the action potential reaches the terminal button, these are the acetylcholine, the neurotransmitters which are stored again in the vesicles. They are released. Once this acetylcholine is released into this neuromuscular cleft, this gap, on the muscle membrane, there are acetylcholine gated receptor channels. These are what you call acetylcholine gated receptor channels. And this acetylcholine gated receptor channels can accept this neurotransmitter, that the acetylcholine. And when the acetylcholine attaches, sodium and potassium gates are open. More sodium diffuses inside, but less amount of calcium goes out. See, when the information impulses comes from the motor neuron, the voltage gated calcium channels are open, so calcium ions flow inside. It makes the vesicles storing the acetylcholine to release them into this neuromuscular cleft. And on the surface of this uh, muscle membrane, there are again voltage gated sodium and potassium channel, sorry, voltage gated uh, acetylcholine receptors. These are the acetylcholine receptors, and this acetylcholine binds to these acetylcholine receptors. And once uh, they attaches, they forward the information that is brought from the nerve ending to the muscles. They forward the information that has been brought by the nerves. And once the they are able to pass the information, they go. So the calcium uptake at the terminal causes the release of neurotransmitter, that is the acetylcholine, into the cleft, and then this acetylcholine travels across the synaptic cleft in the postsynaptic membrane. This is the postsynaptic membrane, also known as motor end plate, on which you have acetylcholine receptors. And motor end plate mainly contains nicotinic receptors for acetylcholine. So acetylcholines are attracted by these nicotinic receptors, which are ligand gated channels. This acetylcholine binds to the alpha subunits of nicotinic receptors, and that causes the conformational changes. When the acetylcholine is able to bring the conformational changes, the central core of the channel opens, which causes the permeability of motor end plate to sodium and potassium ions. So this the same thing, the permeability of the motor in the motor end plate increases. It increases the permeability to sodium and potassium ions. Uh, Open the ligand gated sodium channels as well as the potassium channels. Now this is being caused by when the acetylcholine attaches with the ligand gated sodium channels and make the sodium channels to infuse. As a result, sodium and both the potassium flows down their concentration gradient. This causes the depolarization of the end plate, and this depolarization of the end plate causes end plate potential. End plate potential is not the action potential, but it simply depolarizes the motor end plate. It's mainly because small quanta packets of this acetylcholine is released randomly from the nerve cell, and this acetylcholine, when they attach with the ligand gated channels, they change the membrane potential of the motor end plate slightly, and that is what we call miniature end plate potential. But when large number of such packets of acetylcholine attaches with the ligand gated channels, each causes miniature end plate. Together, it results in large end plate potential. Now, what the end plate potential does? End plate potential spread on the muscle membrane, muscle fiber, and now the muscle fibers get depolarized to the threshold level. That causes the action potential. This end plate potential is responsible for generating what is known as the action potential. In order to ensure purposeful movement, muscle cell electrical response is turned off at this time. Acetylcholinesterase, an enzyme, it degrades acetylcholine to choline and esterase. Once the acetylcholine can pass on the information by generating end plate potential and thereby action potential, an enzyme acetylcholinesterase degrade this acetylcholine into choline and acetate. About 50% of this choline is returned back to the nerve endings. nerve terminal, presynaptic terminal by sodium choline transport. And the same choline is used for reused for acetylcholine synthesis. Now the muscle fiber can relax. Now or the muscle fiber comes to the relax. If sustained contraction is needed means another motor neuron and fluid uh, leads to the release of more acetylcholine and continuously it should that there should be information from the nerve. It should release the acetylcholine and acetylcholine should bring all these changes continuously. That is what we call if the muscle contraction is really needed continuously. So this is how the muscle contraction is brought about. Can you hear me? ಕಾಣಿಸ್ತಾ ಹಲೋ
Yes, sir.
ಸ್ಲೈಡ್ ಕೂಡ ಕಾಣಿಸ್ತಾ ಇತ್ತಲ್ಲ ಕೇಳಿಸ್ತಾ ಇತ್ತು ಓಕೆ ಎನಿಥಿಂಗ್ you want to share with me or if you have any difficulty in understanding the process or if you have any doubt please even you can send chat with me or you can uh, ask now also. Hello. So whatever the things I have recorded I will be able to send it to you again if you want to do this one and if you don't have any problem or anything to share I would like to wind up now. Okay.